Good afternoon, everyone. My name is Edwina Kinchington, and I'm the chair of the Young Women in Pittsburgh group. And thank you very much for tuning into our Young Women in virtual presentation today. We're excited to announce the availability of these online resources through our brand new YWIB online platform. If you have any questions, I'm going to ask that you please hold those questions till our Q&A period at the end of this session. In addition, if you have a burning question that you don't want to forget, you are welcome to put it in the chat. So Young Women in Bio is launching a new initiative for young women who are curious about STEM and who want to learn more but may not live near one of our 13 chapters in the United States or in Montreal. The mission of Young Women in Bio is to inspire our next generation of young women in science and support their need to become tomorrow's leaders in the STEM fields. We strive to provide education and hands-on experiences in STEM, as well as show our passion for all fields that are scientific. Of course, with the time of COVID, most of our activities at the moment have been virtual. The launch of YWIB Online comes at a time when there's a real need to provide young girls who cannot otherwise attend our events and programs and online programming in STEM. This will help our goal to ignite curiosity and inspire young women to see a future for themselves in STEM. We believe that by making these online resources available to a broader audience of our girls, we can encourage more young women to find a lifelong passion and career in STEM. Young Women in Bio is part of the Women in Bio organization with chapters nationwide and in Canada. WIB is a nonprofit volunteer organization that promotes diversity and inclusion for all women in the life science and beyond. WIB partners with leading companies, universities, hospitals, and other organizations to host highly engaging and educational and motivational programs for young women interested in STEM. There are more than 13 chapters across the United States and in Canada. And if you want to learn more, please visit our website listed here. Today, we welcome Dr. Amy Hartman and Dr. Anita McElroy to share their journey of how they became successful scientists in the fields of vaccines. Both investigators work at the University of Pittsburgh. Dr. Hartman is an assistant professor in the Department of Infectious Diseases and Microbiology in the School of Public Health. She received her bachelor's degree in biology from Washington and Jefferson College in 1998 and her Ph.D. in molecular virology from the Department of Molecular Genetics and Biochemistry at the University of Pittsburgh School of Medicine in 2003. Dr. Hartman has worked in the Special Pathogens branch at the CDC, the Centers for Disease Control and Prevention in Atlanta. During her career, she has worked on several highly contagious and infectious viruses, including HIV, Marburg, and Ebola virus. But presently, she runs the Biosafety Level 3 facility at Pitt and the Center for Vaccine Research. Dr. McElroy is a pediatric infectious disease physician, a medical doctor with a research interest in hemorrhagic fever viruses like Ebola and Marburg. She received her Bachelor's of Science from the University of Maryland, her PhD from the University of California in 2000, and her medical degree from George Washington University in 2007. Presently, Dr. McElroy provides clinical care at UPMC Children's Hospital of Pittsburgh, and her lab focuses on the mechanisms by which hemorrhagic fever viruses cause disease. Dr. McElroy and Dr. Hartman, thank you very much, and I will pass it over to you.
Okay, thank you for that introduction. I'm going to go first, and I will tell everybody a little bit about my career path, and then Dr. McElroy will do the same, and then we will start talking a little bit about vaccines, what they are, and what they mean for the current COVID outbreak. So I just want everybody to know that I'm happy to be here talking to you all. If you have any questions, please let us know. I'm happy to answer. As Dr. Kinchington said, I am an assistant professor in the Center for Vaccine Research, and I'm in the School of Public Health in the Department of Infectious Diseases and Microbiology. And so I always like to start out, especially a local talk like this, telling you all that I am a native Pittsburgher. I graduated from the Woodland Hills School District. So I always just like to remind local Pittsburgh kids that that you can go on and do the types of things that we do. From there, I went to Washington and Jefferson College, just about a half an hour south of Pittsburgh. I was a biology major, a Spanish double major, and I graduated in 1998. And then I came here to Pitt, and I started graduate school in what was then called the Interdisciplinary Biomedical Graduate Program. So this is a program within the School of Medicine where you can earn a Ph.D. In the particular program that I was in was molecular virology. And so I earned that degree in 2003. And from there, I went to CDC, which is in Atlanta, Georgia, and I did a postdoctoral fellowship. And that's where my path crossed with Anita, in that we both kind of came from the same place here in the CDC. I worked during my time there in this viral special pathogens branch. And that kind of brought me full circle back after CDC. I came back to the University of Pittsburgh about 11 years ago now. And I took a position in the Center for Vaccine Research at the time as sort of the manager of the high containment research lab there. I'll tell you a bit about that in a minute as well. And so that's my career so far. And just to tell you a little bit about my time in Atlanta at the CDC, which is known as the hot zone, because this is the lab, the high containment lab where we work with dangerous pathogens like Ebola. And I got a lot of really good experience working in the high containment lab, as you can see from these pictures here. Here I am in a couple of different types of what we call spacesuits or protective gear that we wear to protect ourselves from what we're working with in the lab. These are completely self-contained suits. So they're sealed from the environment, and air is provided to the suit through the red hose. And so you can see that we have regular lab equipment, but we're wearing these high containment type of protective equipment. And during my time at CDC, I traveled to Africa, to a country, Angola, here on the West Coast, during an outbreak of a hemorrhagic fever virus called Marburg hemorrhagic fever, which is a close cousin of Ebola. And during my time there, I was located in the capital city of Luanda, where we set up a diagnostic laboratory to take patient samples, to test them for the presence of the virus, and then report that information back to the health authorities like the World Health Organization. So here are some pictures from my time there working in the high containment lab that we had set up, and then extracting nucleic acids and running the diagnostic test. The test, which is not unlike the test that we now have for COVID-19. And so now, being back at the University of Pittsburgh, my experience at CDC working in the high containment lab there gave me the experience I needed to come back here to Pitt and help establish a biosafety level 3 facility, which is one step down from the what I showed you before in biosafety level 4. So this is one step down, but it's still a high containment facility. And these are some pictures of what the type of protective equipment that we wear in a BSL-3 facility. And so I came back here and oversaw the opening of the facility. And now since about 2014, I run my own research laboratory focusing on emerging infectious diseases such as SARS-CoV-2, which causes COVID-19. And so Dr. McElroy and I both work in the Center for Vaccine Research. So a lot of our work is geared towards vaccines for emerging infectious diseases or therapeutic interventions. And so, as you would imagine, when COVID-19 happened back in January and February, we formed part of the center's COVID-19 response team, where we obtained early isolates, set up some initial animal models. So a number of different animal models that are needed for testing the vaccines that you hear about in the news. And that's what we're working on now. So we, both of our labs, and she may tell you a bit about this too, but both of our labs have been working in this area, really trying to learn all we can about this new virus and set up the tools that are needed to advance vaccines and therapeutics. So if you have any questions or if you want to find out more information about what it is we do specifically at the Center for Vaccine Research, you can visit the website, or you can look at my lab website and you can find out a little bit more about what I do. So I'm going to stop there and I'm going to turn it over to Dr. McElroy, and she'll tell you a little bit about herself.
All right, very good. Are you seeing my slides?
Yes.
Okay, perfect. So thanks, Dr. Hartman. Just following up on what we just discussed. I have a relatively similar background to Dr. Hartman, except I took a little bit of a detour, which I'll go over in a second. So I in fact wear two hats in the job that I have right now. I am a physician and I'm a scientist, and I've listed on this slide a few things to help you sort of understand what the difference is, because if you're looking at a career of either the physician or the scientist, one may appeal more to you than the other, or you may, like me, think, wow, I want to do both of those things. That's a lot of fun. So the physician's job is really to observe signs and symptoms in their patients. They order laboratory or other studies to figure out what the problem the patient has, and then they come up with treatment modes for the patient. And they're really oriented on understanding what is the disease process, like what is the problem and how do I fix it. But the scientist thinks about things in completely different ways. So scientists observe patterns and association. They look at what they observe and then they make hypotheses about why something might happen a certain way. And then they design experiments to test out those hypotheses. And they don't just ask, how do I fix it? But they ask, why does it happen? So these are really two different ways of thinking. And as a scientist, I do that work in the Center for Vaccine Research here at the University of Pittsburgh. But as a physician, I'm a pediatric infectious disease doctor, so I do that work over at UPMC Children's. And so similar to Dr. Hartman, here's my timeline. I'm showing you here that I was born in 1975, so you can do the math if you want to know how old I am. And there's a movie that came out in 1975 that you may or may not have ever heard of, and we used to play movies on tapes that look like this, and if you've never seen one of those, that gives you a sense of just how old I am. So I graduated from high school in Maryland in 1993, and then I went to college at the University of Maryland College Park, where I studied microbiology. Then I went to UC San Diego, and I got to live in beautiful Southern California for 4 years, where I got a Ph.D. in biology studying herpes viruses. And then I went and did what's called a postdoctoral fellowship. This is when you're a PhD level scientist, but you want to really expand your area of study in a slightly different area. And I wanted to study emerging viruses. So I went to a place called USAMRIID, or the U.S. Army Medical Research Institute for Infectious Diseases. And that's where I did my postdoctoral fellowship. But while I was there, I realized that I wasn't content to be doing only science, that I really wanted to have a little bit more direct one-on-one impact with people that I wanted to be able to help. And that led me to the medical degree. So I went back to school and I got an MD at the George Washington University. But getting an MD is not the end. So it's important to know if you're thinking about going to become a physician, you get an MD, that's a 4 years of training, but then you have to go and get specialized training in the kind of MD you want to be. So I knew I wanted to be a pediatrician. So I went and did a pediatric residency, and I did that at Emory University. And that was from 2007 to 2010. But I also knew I wanted to be a pediatric doctor who specialized in infectious diseases. And in order to do that, you do what's called a fellowship. So that's another 3 years that I spent from 2010 to 2013. I also did that at Emory. So that's 4 years of college, 3 years of residency, and 3 years of fellowship to get you to the point where you're ready to practice medicine independently. Not everybody has to go down all those routes, and I'm happy to answer questions if people have them about the details related to this. And while I was at Emory doing this training, I did all of my research. I continued doing science, and I did that research in the viral special pathogens branch at CDC, which is where Dr. Hartman and I had met each other because we overlapped a little bit there. I stayed on at Emory for several years from 2014 to 2017, where I had my first real job. So you can imagine I was in my 30s by the time I had my first real job, which is totally fine. And I was super happy I got to spend all those years studying. Then in 2017, I was recruited here to the University of Pittsburgh, where I get to wear both of these hats, right? I get to do both of these jobs by working at UPMC Children's as a pediatric ID doctor, and I get to do the science stuff working at the Center for Vaccine Research. So I want to just show you a few slides about things I've been able to participate in being either a doctor or a scientist, and just tell you a few stories that I think are fun things that I got to participate in. So as a physician, you get to meet famous people. Here I'm showing you that Mickey and Minnie came to the hospital, but we often have people from the Steelers or the Pirates or the Penguins come over to UPMC Children's to see the kids. So it's always a fun place to be always fun to be a pediatrician. The other thing that I've had the opportunity to do as a physician is to travel to other parts of the world where there are folks who don't have ready access to medical care. So these are some pictures from a trip I took to Haiti many years ago. And you can see here I am on the left, I'm just interacting with a young gal who has a head that seems significantly larger than you would expect it to based upon her body size. And that's because she has a medical condition called hydrocephalus, where fluid accumulates on the brain. And without surgery, there is no controlling the size of those heads, and they can get bigger and bigger and bigger. Here I am on the right doing some dental work on an individual who had a rotten tooth, and you can see the rotten tooth there in my hand. Forgive me if these pictures are a little too graphic for some of you. This is a really important thing in Haiti because people don't have regular dental care, and they often chew on sugarcane, which grows naturally there, and they get a lot of cavities, and they're not able to have regular good dental hygiene. So brushing your teeth after eating is very important. Here on the left, I'm examining a little girl that we met who has a cardiac disease. So that means her heart is malformed. And because of that, her liver is bigger because of the heart not being able to function correctly. So one of the things that we were trying to do there was to identify folks who needed referrals to specialized medical care. And this little girl was one of the ones that we were able to refer, in addition to the little girl here with the hydrocephalus. And then here on the bottom right, you can appreciate this, this young lady has this really cool, fun, spiky red hair, but her problem wasn't that she had a nice hair color, but her problem was that she had nutritional deficiencies. So she had both skin and hair abnormalities that were related to her lack of access to good nutrition. And so we were able to get her involved in a program to basically help beef up her nutrition. So these are really fun, rewarding experiences to be involved in. I want to tell you about two other cool clinical cases. This is a teenager who has a cancer, a leukemia, and is on a lot of medicines to treat that leukemia. And those medicines have made her immune system not work so well. And so she came in one day with this huge knee, right? This knee this is not a normal looking knee, right? And I went to the operating room with the physicians when they cut her knee open. And so you can appreciate they've cut open the knee here, and all of this yellow pus is oozing out of her knee. And the really cool thing about this is because I was there in the operating room, I was able to take a sample right away. And because I'm a scientist, not just a clinician, I took that sample back to the lab and I stained it with something called acridine orange. That's the picture down here on the right. And that's a dye that stains nucleic acids. So the DNA and RNA that lives in our cells will be stained by this. And you can see cells from the patient here in the bottom right that are staining. That's the nucleic acid of the patient. But then you can also appreciate the nucleic acid of this microorganism. This turns out to be a fungus called aspergillus that was causing her infection. So that very same night that she had this procedure, we were able to put her on the right medication because we were able to immediately go to the lab and look and figure out what this was. Another fun case that I like to tell people about is this little guy. So you guys are all from Pennsylvania, so you're familiar with ticks, but this is a slightly different kind of tick disease, and it's called tick paralysis. And this happens when a tick that binds to you produces a toxin that basically makes your nerves not work. It's a very rare disease, but this 6-year-old boy, he came in with weakness of his legs to the point where at first he was just wobbly, and then he couldn't walk, and then that weakness got worse and worse to where he couldn't breathe. And his diaphragm, that's the muscle that helps you to breathe, wasn't working. So he had to be put on a breathing machine in order to breathe. And he's actually on that breathing machine here in this picture on the right. right? And I'm just going to play you this cool video because this is Dr. Miller demonstrating how to remove a tick.
Parts. And then you want to pull them straight out gradually. You try to tent the skin.
Somebody's bringing me a container.
And usually they relent.
So we can use this for right now.
Look, they're taking the stick off my kid's head.
Parts.
So that tick was really stuck into that guy's head, which was a real problem. But we got it out and he was totally fine afterwards. Within a couple days, he was running around the hospital and got to go home. So it was a very rewarding case to be able to cure someone just by removing a tick from them. And then I want to tell you one last story about things related to Ebola. So just like Dr. Hartman had the opportunity to do some work in Africa related to Marburg, I was fortunately in the right place at the right time when I was working at Emory and working at CDC when, if you'll recall, in 2014 to sort of 2017, there was an outbreak of Ebola. This is a very severe infection. It kills a lot of people when they get infected. And some of those patients who had Ebola were brought back to the United States and cared for at Emory University in Atlanta. This is a picture from CNN when one of the first patients came in. This is another picture when another patient came in who was actually a little sicker and had to be wheeled in on a gurney. But because I was a research scientist studying Ebola just down the street at CDC, I was able to work with my colleagues at Emory and CDC, and we learned a lot about immune systems and how the human immune system responds to infection with Ebola. And I'm just showing you the titles of several of the papers that we all published together that was a basis that came out of the work that we were doing there. So this is really applying the scientific mind to the clinical human disease. And then because I'm a physician and a scientist, there was a scenario in Guinea that I was able to participate in which this little girl right here, Nubia is her name, and she's publicly known, so I can show you her pictures. And Nubia was cared for by the doctors of MSF, Médecins Sans Frontières. And these guys, it's their job to care for patients with Ebola. And Nubia's mom came in with Ebola disease and was very, very sick. And Nubia's mom actually died while giving birth to her. And no baby had ever survived being born to a mom that had Ebola ever before. And so it was a really high-risk situation, and no one thought that Nubia was going to make it. And we together worked to give her some experimental therapeutics, things that hadn't yet been tried and true and proven. And Nubia miraculously survived. And so you can see her when she's leaving the ETU, all better, and then here at a follow-up checkup point, and then here with her aunt years later. So it was a very rewarding thing to be able to participate in. So I'm going to stop sharing that, and then I'm going to try and share with you a different slide set. This is the one that Amy and I have put together to talk with you about vaccines. All right, there we go. Can someone just let me know that they see these slides okay?
Yes, thank you. And thank you both Dr. Hartman and McElroy for that background. It was very interesting.
We hope that you guys find vaccines just as fascinating. So I wanted to start by basically throwing out a few questions here, and I'm hoping that folks can pipe up so you can unmute yourself and just say a few things. We'll see how well this goes. So the first question is, what is a vaccine? Well, I'll tell you the answer, right? I've given you an answer here. It's a treatment that when you give to someone prior to exposure protects them from an infectious disease. And I'm sure that you all have heard of some vaccines. So I'm going to invite you now to just throw out the names of some vaccines that you might have heard of. If you want, you can put them in the chat, and then maybe Dr. Hartman could say them out loud, because I can't see the chat.
I can help you with this. So our participants are saying flu shots, polio vaccine, TDAP,
Tdap, yep. Good.
chickenpox.
Chickenpox. Excellent.
We
So,
have a flu shot again, rabies,
rabies. Good.
MMR.
MMR, yep. All right, you guys know your vaccines. That's really impressive. So does anyone in the audience know anyone who's ever had smallpox? What about anyone who's ever had polio? Does anyone know a person who's had polio?
From the audience, it seems no one.
Okay, what about measles? This one is maybe possible. We had a measles outbreak here in Pittsburgh not too long ago. Does anyone know anyone who's ever had measles?
There is one person that actually has a connection with someone who got polio.
Okay. So why do you think it is that most of you have never met anyone who's had these diseases?
You're welcome to share, either unmute yourself or in the chat.
I think also because a lot of the vaccines have been created like to help prevent these diseases from coming more often.
Yep, that's exactly right. So the reason you don't know anyone who's had these diseases is because we have vaccines that protect us from these diseases. So in some ways, we're the victims of our own success, right? We've forgotten about these things because we don't realize how much disease and damage that they have caused in the past. Here's a table showing you what the consequence of introduction of vaccines has been on disease. And smallpox, no one should have ever met anyone with smallpox because smallpox has been eradicated since the 1970s, right? This virus does not exist in humans on this earth anymore because of a massive vaccine effort that went out around the globe to prevent this disease. And because people are the only ones who get smallpox, we were able to eradicate this. So the disease has been 100% decreased. It has essentially been eliminated. This data that you're seeing here is actually US data on this particular chart. You can see that we don't really see diphtheria in the US anymore. We rarely see pertussis. If anyone's interested, we can talk about why we still see some pertussis cases. We rarely see tetanus. We never see polio anymore, right? So this is the legacy of the Center for Vaccine Research, right? Jonas Salk was a researcher doing science here in Pittsburgh and developed the first polio vaccine. And we went from having 20,000 people a year with paralysis related to polio to nobody. It's a really amazing success story. Measles we've talked about. We hardly ever see measles. We only see measles occasionally now because there are some folks who neglect to get their vaccine and so they're not protected. So let me show you just another way to look at this same information. This is just a graphical way to look at it. There's a few other vaccine preventable diseases listed here. So hepatitis A and B are also on here, as well as mumps, and then something called pneumococcal disease. This is what causes pneumonia. And you can appreciate that before vaccines, there were a lot of cases of all of these diseases. And then after the institution of vaccination programs, we see massive, massive reductions and in sometimes complete elimination of these diseases. So vaccines really do work. Here's two examples of how those vaccines work. So these are cases of paralytic polio. This happened in 1 of every 2,000 people that got infected, and that resulted in basically 20,000 cases a year in the 1950s. And here in 1955, when Jonas Salk introduced the first polio vaccine, you saw a massive decline, and then there was a second version of that vaccine that came out. And then by the late 70s, we didn't have polio in the United States anymore, right? It's a huge success story. We're really, really close to eradicating polio in the world. There's only one country left where we have just not yet gotten complete vaccination. So stay tuned. That will be another success story. Measles is another fantastic success story for vaccination in the United States. So, 500,000 cases every year. This is a fever, rash, respiratory disease kind of illness. It's the most contagious disease on the planet, right? More contagious than anything else. If one infected person can infect 15 other people, but by instituting the vaccine, you see a dramatic reduction, and we rarely see cases now, except for the problem we have when people refuse vaccination. So I want to just give you a little bit of information on how vaccines work, and then Dr. Hartman's going to take over and talk about COVID vaccines specifically. So vaccines work by stimulating your body to generate an immune response against a disease. And what happens is this then protects you when you get exposed to the real thing. This is a somewhat complicated slide, but I don't want to oversimplify this. This is really complicated stuff. So when you give someone a vaccine, the stuff that is in the vaccine, we call those antigens, right? And those antigens, they're usually proteins. Those proteins are picked up by specialized cells in the body called antigens presenting cells. And that's exactly what they do. They take that antigen and they show it to other immune cells like B cells and T cells. And those B cells and T cells then work together to make antibodies. And those antibodies can attack a virus and prevent that virus from causing an infection. The immune system also produces cells called cytotoxic T cells. And these are cells that can recognize infected cells and release things that kill those infected cells. So we have two major arms of our immune system that are important following vaccination. This is how vaccines work. The humoral arm, which is the antibodies, and the cellular arm, which is the T cells. And then there's four major classes of vaccines, four different types of vaccines. So one is called a live attenuated. So this is just a weakened version of the real thing. So this is a real pathogen. It can replicate, but it's weak, so it doesn't cause disease. And the measles vaccine is a good example of this. We also have killed or inactivated vaccines. So we take the real thing and we treat it to basically kill it and make it non-infectious. And the polio vaccine is a good example of that. And then we have subunit vaccines where we take a little portion of the pathogen, a little protein, and we show it to the immune system. And then the immune system recognizes that when it comes in contact with the real thing. And the hepatitis B vaccine is a good example of a subunit vaccine. And then finally, and Dr. Hartman will probably mention these, there's this idea of a nucleic acid vaccine. So this is the genetic material, the DNA or the RNA, little tiny bits of that from a pathogen, and it encodes a viral protein. So when you put the nucleic acid in a person in the form of a vaccine, the person's cells turn the nucleic acid into protein, and then the body recognizes that protein as foreign and generates an immune response to it. We do not currently have any licensed vaccines that are based upon nucleic acids. But there are some in the works. So I'm gonna stop and leave this slide up for a second, and I'm gonna stop sharing, and Dr. Hartman's gonna take over and share her slides and talk about the rest of this.
Okay, can you see the SARS-CoV-2 map? All right, well, Dr. McElroy gave you a nice introduction of what vaccines are and a little bit about how they work. And so we find ourselves here in 2020 in the midst of one of the largest global pandemics of any infectious disease. SARS-CoV-2 is the virus that causes COVID-19, which is the disease. And so, as we all know, we need a vaccine for this. And the reason is because all of us are sitting at home on Zoom looking at this presentation because we can't be in the same room together. And so we all want to get back to our normal lives, whatever normal is going to look like after this. But we all want to get back to being able to go and do things, go to restaurants, go to schools, go to ball games. And so we need a vaccine. And so the latest statistics on COVID-19 are shown here on the left. So Johns Hopkins has this really nice online portal where you can get a real-time look at the number of cases. And so globally, there have been over 38 million cases of COVID-19 with over 1 million deaths across the world. In the United States, the map looks like what's here on the right side of this slide, where we've had close to 8 million cases and over 200,000 deaths. And so if we look at sort of the curve of cases over time, we can see we had this peak in the spring when we were all on lockdown. It went down a little bit, but then came back up in the middle of the summer. Remember when things got really bad in July and August, they went back down and they're starting to creep up again. So we don't know whether we're in the middle of a third wave here or what's gonna happen when winter comes. But what we do know is that the sooner we can have a vaccine, the sooner we can get back to some semblance of normalcy. Now, I want to talk a bit about what it takes to develop a vaccine. This is not a simple process. Dr. McElroy talked about the immune response that you have to generate. Well, in order to be able to design a vaccine that can protect people, you have to understand a lot about how the pathogen causes disease. and this takes time. And so this slide that I have up here shows you what the traditional vaccine development process is. And this takes years. If you look at the bottom here, you know, it can be anywhere from 5 to 15 years to develop and license a vaccine. So most of the vaccines that we have now have been developed in this pathway. We obviously can't wait 15 years for a COVID vaccine. So I just want to go over some of the steps that have to happen in order to get a vaccine that you can give to a person in a clinic. And so a lot of the time is spent in the beginning here doing basic research, what is listed here as target or discovery and target validation. So this is where basic scientists like myself and Anita, we do research in the laboratory to try to understand how these pathogens work, how do they cause disease, how do they infect cells, how can we design a vaccine that can prevent that, you know, what is the immune response in animals. So all these types of experiments are done very early on in the discovery phase, and that can take years. Now with COVID-19, this has all been compressed into a very few short months, but we're still at this stage where we're learning about how this virus causes disease. Once we get a little bit of an idea of how it works, how it causes disease in animals, and potential ideas for a vaccine, then you can enter what we call the preclinical stage, which is where a vaccine candidate is tested in a very controlled manner in animals to try to really understand the immune response that's generated in animals and how the vaccine can protect them from challenge. So all of this early stage takes place way before any vaccine ever gets into a human being. Once the data from this early stage looks pretty good and pretty promising, then a vaccine candidate can move into phase 1. So there are 3 phases of clinical trials in human beings. Phase 1 is called the safety phase. So this is where the vaccine is given to a very small number of people initially, just to make sure it's safe, and safe in a very general sense, right? So it's given to a small number of people, make sure there are no major issues. And then the vaccine can enter phase 2, which is both safety and immunogenicity. So here it's given to maybe a couple 100 people, where you're still monitoring safety very closely, but you're also monitoring the immune response that's generated and comparing that immune response to what was learned back at the beginning in animals and seeing if they're comparable. It's during the phase 1 and phase 2 where the dose of the vaccine is tested to try to find the optimal dose that you would use to give people. And then the third phase, which is you know, arguably the most important phase, is when it's actually tested to see if it can protect people from the disease. So phase 3 is safety and efficacy. Efficacy means, does the vaccine work? You know, if a person takes this vaccine, are they less likely to get the disease than if they don't have the vaccine? That's what efficacy means. And once all 3 phases are complete, then regulatory review happens. So this is where an agency like the Food and Drug Administration, the FDA, they look at all the data. They look at the safety, they look at the immunogenicity and the efficacy, and they make a determination whether, yes, this vaccine looks like it works and it's safe, we can license it for use in people. And that's just like a sort of a simplistic overview of how the process happens. But generally, these steps occur sequentially. But because of the situation with COVID-19, we can't wait this long to develop a new vaccine. So if you read in the news and hear about what's happening with the current vaccine candidates, some of these phases have been overlapped and kind of combined together to sort of accelerate the process, right? So while those of us like Anita and I were you know, over the months of the shutdown working on this early stage trying to understand this virus, there were companies that were already starting using known vaccine platforms, already starting early phase 1 trials. and they've sometimes combined the phase 2 and phase 3 together. So there have been a lot of ways to try to condense this, but it's generally speaking a very long process. And if you go to the New York Times has a really good coronavirus vaccine tracker, which they update pretty much daily. And it tells you what are all the different vaccines are and where they are in the process. We have at least 44 vaccines in human clinical trials right now. And then a whole lot more that are in the preclinical phase where they're still under investigation in animals. So right now we don't have a vaccine that's approved for use in humans here in the United States, but we've made tremendous progress in the 7 or 8 months since the outbreak started. And this has been the fastest that we've ever had a brand new virus where we've learned a lot and proceeded towards a vaccine this quickly. I'm just going to mention briefly a couple of vaccine candidates that you might hear about in the news if you listen to the news when they update on COVID vaccines. The federal government has started this Operation Warp Speed to try to roll out these vaccines very quickly. I want to mention the first two here that Dr. McElroy mentioned, the idea of an inactivated vaccine. This is like kind of the simplest way to design a vaccine is to take the virus, inactivate it or kill it, basically make it so that it's no longer harmful. And you take that inactivated virus and inject it into people. And when your body sees that, it sees the proteins from the virus and it generates an immune response. There are a couple of these inactivated vaccines that are in the pipeline. And from what I can read and what's been published so far is that some of them have been approved for limited use in China, but none in the United States at this point. And then we have a number of vaccines that are what we call vectored vaccines. So this is where you take sort of a vector that will shuttle the protein from SARS-CoV-2 into the human body. And a lot of these are using another virus called an adenovirus. And so an adenovirus is a virus that is commonly found in people, is generally not harmful, and you can take a weakened form of the adenovirus and insert one of the proteins from SARS-2, and then this can serve as the basis of the vaccine. So there are a number of, actually at least 3 candidates that I have listed here that are like attenuated vaccine candidates based on adenovirus. And a number of these were just in the news recently. So the one here on the bottom from Johnson & Johnson, you might have heard about that in the news, I believe it was this week. The clinical trials with this vaccine have been paused due to a potential adverse event in one of the patients. So a patient that was given the vaccine as part of the trial developed a serious reaction. They don't know at this point whether it's from the vaccine or not, but they paused the trial till they can look at all the data and figure that out. And then you may have heard also of what's referred to sometimes as the Oxford vaccine. It's made by AstraZeneca, and this is a variation of the adenovirus type vaccine. It also is on pause in the United States due to a potential adverse event in part of the European trial. And then the last 2 vaccines listed on here are nucleic acid-based vaccines. So this is where the vaccine can, instead of injecting a protein into somebody, they're injecting a piece of nucleic acid called RNA. And when you inject that into somebody, then your cell's machinery see the RNA and they convert that to proteins based on the RNA sequence, and then your body can generate an immune response on that. And this you might have heard of in the news. One of these is from Pfizer and one of these is from Moderna. As Anita mentioned, we've not had a vaccine like this licensed for human use. This would be sort of a new technology. And so the question that everybody always asks is, all right, when are we going to have this COVID vaccine, right? Because we all need it, we all want it. When when is it actually going to be here? And the answer to that is, it's not easy to answer because it depends on a lot of different things, right? I don't think we are going to wake up one day and say, the vaccine, it's here, finally, we've got it. That's not how it's going to happen. It's going to happen over time. It's going to be rolled out over time. And there's a high likelihood that we will have several vaccines, and they're going to work in different ways, and they're going to be recommended for different people based on different things. So what makes it so hard to generate a vaccine is, once you go through that whole long process that I mentioned about getting FDA approval, even when you get to that point and you have this great vaccine, there's a huge issue in scaling up the manufacturing so that you can make enough to have enough for everybody, in this country and potentially the entire world. So scaling up vaccine manufacturing is not easy, it's not fast, and it's not cheap. And so part of the sort of overlapping of the steps that I mentioned is that a lot of vaccine manufacturers have been trying to scale parts of their manufacturing machinery up early before we even have a vaccine candidate to make, so that they have everything sort of ready to go. But that's not going to be a trivial part of this. And then there's the issue of, okay, how do you, if you have a lot of vaccine ready to go, how do you distribute it to people, right? How do you actually get it from the vaccine manufacturer to all the clinics around the world where, you or I could go to the clinic, sign up, stand there and have a nurse inject it into our arm? Like, how do you get to that point where it's distributed? That also is not an easy feat. And we can use infrastructure that we have in place, like for the flu vaccine, to help with that, but it's not going to be a simple thing to do. And then there's the issue of, okay, how do you prioritize who receives the first doses? Because whenever we have a vaccine or several vaccines that can be given to people, we're not going to have it all at the same time. So we're going to have to prioritize who's going to receive the first dose. If anybody wants to chime in, who do you think should get the first dose of vaccine that rolls off the lot? Does anybody have like an opinion? Grandmas, yep, Anita said grandmas, right? Yep, that's a good point, right? Elderly people, really susceptible to severe disease. So I would want my grandma to get vaccinated. What else? Does anybody have, yeah, immunocompromised patients? That's a good one, right? Healthcare workers, yeah, exactly, right? So I was thinking the healthcare workers, the nurses, the doctors who have to be around a lot of patients, they should get it early, right? What about people that have like pre-existing conditions that make them prone to having more severe disease, right? So, lots of different conditions. Yep, teachers too. So there's a lot of sort of susceptible people or frontline workers that really should be the ones to get that and to prioritize that. That can also, also be, a bit of a tricky situation, because who can determine who should get it first, right? And so there may be some arguments on who should get that. So I wrote this down, like, right, people that work in the grocery store, they maybe should get it too. People that work in nursing homes, high-risk individuals, like those with asthma, diabetes, cancer. So my last slide here is just to point out that we all want a SARS-CoV-2 vaccine as quickly as possible. And we really have to balance speed with which we get a vaccine out there with the very important aspects of making sure that it's safe and that it actually works. We would never want to administer a vaccine that is not safe or that doesn't work, that would make people doubt the usefulness of getting vaccines, because as Anita pointed out, they've been so tremendously life-changing in the human population over the past 100 years. So I think that's my last slide. We're happy to take any questions regarding anything that we discussed today. All right.
Thank you very, very much, Dr. Hartman, Dr. McElroy. I learned a lot, and we do have a few minutes for questions. You are welcome to unmute and ask your question. You do not have to turn on your video, or if you want to put it in the chat, that's fine. I think you did a great job of concisely explaining kind of how vaccines are developed and the challenges. And thank you for summarizing the COVID vaccines. So I did recently hear about the ones that have been halted. It was unfortunate.
So I will start with one question. First of all, thank you to the both of you, Dr. McElroy and Hartman. This was very interesting. You mentioned side effects, so I have a few questions about that. Can you discuss a little bit which are the side effects? And I have a very specific question. So also the technology delivering protein through viruses, so adeno in this case, is very new to my knowledge. The only, again, to my knowledge, approved is the Ebola vaccine, approved by Johnson & Johnson. Do you think that this vaccine is gonna increase normal side effects that we see on other type of vaccination?
Anita, do you want to talk about side effects, maybe?
Yeah, happy to do so. So the typical things that we worry about as side effects following vaccination are usually local reactions. By that, I mean redness and swelling and pain at the injection site. Those are very common side effects. We've all gotten our flu shot, right? And I hope we're all going to get our flu shot this year. So we know it's not always the most comfortable thing, but it's worth a little bit of pain and discomfort. Every once in a while, vaccines can have more severe side effects. Sometimes when you get your vaccine, you feel kind of yucky for a couple of days, right? You feel kind of down, you feel kind of, don't have the same level of energy. That's a sign that your immune system has recognized that this thing is foreign and is trying to fight it off. So these side effects, we call this reactogenicity, right? That's the reaction that your body has to the vaccine. And that directly correlates often with immunogenicity, i.e. how well your body recognizes the antigen and makes a good immune response. So it's a trade-off, right? Like I'm willing to feel crappy for a couple days if I can now have a vaccine that's gonna work and protect me from COVID. Sarah asked about the Ebola vaccine. So that's a fantastic vaccine. It is a live attenuated vaccine. It is a vesicular stomatitis virus vectored vaccine. So Dr. Hartman talked with you about live attenuated vectored vaccines. And that vaccine has a lot of reactogenicity. I got that vaccine, right? I didn't feel good for several hours. I was on the couch feeling kind of yucky. But you know what? I have fantastic antibodies against Ebola now, and I'm very happy to have those.
Thank you. There are a couple of questions from the participants also. How can we know that politics didn't influence the approval of the vaccine?
Yeah, that is a good question. And I think in order to make sure that, we can't really control whether politics influences the approval of a vaccine, but what we can do is do our homework and be educated on the vaccines that are licensed, right? So, I said there's probably going to be several different types of COVID vaccines, and we as individuals would probably want to talk with our doctor to understand how each one works and how each one is different. The idea of there being a licensed vaccine before the election in a couple of weeks is just not realistic. Maybe the timeline in my head that the earliest that you could get tentative approval for a vaccine, maybe here in the winter, like February, but a tentative approval doesn't mean it's ready to go in everybody's arm, right? I would say if we're able to get a COVID vaccine like the end of next summer, that's doing probably really well. Maybe Anita feels differently, but we can't change like the political environment right now, but we can talk to our doctors and sort of educate ourselves on what is out there.
On that note, there are kind of similar questions regarding should we be worried about the fast pace of the vaccine production? Are you guys worried about that? What do you think?
Anita, do you want to take that first?
Yeah, sure, I'll take that one. I'm not worried about the fast pace. Here's the difference between the way it's traditionally done, like Amy had described to you, and the way it's being done right now. It's money. That's the bottom line. It's about resources. It's about people to do the work. It's about manufacturing of the vaccine. It's about getting all of the regulatory things in place. There are so many different parallel things that have to happen in order for a vaccine to go through that process. And if you throw enough money at it, which is exactly what has happened in Operation Warp Speed, you can speed up a lot of those different phases. And the other thing that we've done in this outbreak, in this particular scenario, is we've taken advantage of information we already knew from decades and decades of experience in understanding viruses and vaccines and immune responses. And we've used all that information to apply to this particular scenario. And I don't envision there being a problem with applying the same kinds of resources and intellectual motivation to any vaccine. The limitation is resources. It's all about, is there enough money and staff and all the things that take to actually go through the process of doing these trials. And the other thing that Amy mentioned is the fact that we have things happening simultaneously, which normally, like she said, they happen one, then the other, then the other. We do phase one, and as soon as we have the phase one data, we're doing a phase two. I mean, it's not, there's no waiting for something to be evaluated. So I think it's feasible to do this fast. It just takes a lot of money.
Yeah, and one other thing I'll add to that I forgot to mention on my slide with the time frame is that even after a vaccine is licensed, there are a lot of follow-up studies that are done. So the safety and the efficacy of that vaccine is monitored for a really long time, even after it's licensed. It's not like when it's licensed, that's it, they're done. They keep gathering data, they keep looking at efficacy, they keep looking at safety, because sometimes there may be safety issues that don't pop up till really long down the line. So the issue with developing a vaccine very quickly, we can definitely do it, you just don't have as much time to monitor the patients after vaccination for potential side effects. That's one of the issues.
And just one more question that I see in the chat is, how do scientists actually decide who gets a vaccine first if there's limited manufacturing?
Nita can answer that. She's actually
it looks like the question is about in the context of a clinical trial. That's what it looks like in the question that I'm seeing. So basically, vaccines are first tested in adults, right? And ideally, you want to test your stuff out in a normal, healthy adult population. People who don't have immune system problems, people who don't have other major medical problems, because that's sort of the optimal scenario. And so the standard way to go about testing a vaccine is to test it in normal, healthy adults first. And then you test it in older adults, because older adults often have harder times with their immune response. And that's been done for several of the COVID vaccines. And then usually you get to the point where you start to test your vaccine in children. And just yesterday or today, there was something in the news about one of the vaccine manufacturers having approval to test their vaccine now down to age 12. So all this is happening, again, at lightning speed and simultaneously.
Great. So we're pretty much at our time at the moment. I know everyone is busy. I want to thank our speakers, Dr. McElroy, Dr. Hartman, and all of our participants for attending. This recording, if you want to listen to it again or share it with others, will be posted on our YWIB online. And at this time, we'll go ahead and end. Thank you very, very much. We're most appreciative, and I learned a lot today. I hope other people did too, about how you got to where you are and about vaccines and the COVID vaccine approval process. So thank you very much.
Thank you. Our pleasure.
Thanks, everybody.